Multiple voxel pattern analysis. Okay, so I've talked about this briefly before, but I'm going to go through it again with a face example. Just one slide here and we'll be done. So remember, when I talked about it before, I raised it more as a challenge that Haxby and his colleagues were raising about the specificity of the face area, because they were using this method to show information about things that weren't faces in the fusiform face area. And that's true, it's the same method, but this is a way to use that method to just, you don't need to address that debate. You can just use it to say more generally what information is represented here. What discriminations can this part of the brain make? Okay, so now let's consider how to use this region to look at individual face identity. Okay, so what you do is you scan subjects while they look at a bunch of categories. In this case, we'll have two. We'll have Julia Roberts and Harrison Ford. This is actually an experiment we did in my lab a bunch of years ago. Subjects went in the scanner and spent 2 hours being scanned on a block design, looking at just lots of different pictures of Julia and lots of different pictures of Harrison, a real party. So we split the data in half, and here is the pattern of response shown abstractly in the fusiform face area when you look at Julia in half the data and when you look at Harrison in half the data. Okay. And then in the other half of the data, we have patterns as well. And the whole point of this is to say, if the identity of the face is coded by the pattern of response across those voxels in that region, then we should get a stable pattern for an individual that's stably different from the pattern you get for another individual. That's the whole idea. That's all there is to it. And the simple way you can operationalize that, you can do it with other fancier methods, but the simplest way is to just ask, are patterns more similar within a category? If you just correlate those two, Julia even runs to Julia odd runs, is that a more similar pattern? And Harrison even to Harrison odd. Are those more similar than the between categories? Julia even to Harrison odd and Harrison even to Julia odd. Okay? And so basically, if you find a higher correlation within a category, where here a category is just individual identity, or sex, or God knows what else, right? If that correlation is higher within than between, that tells you there's information in there in that pattern of response. Does everybody get that method? Okay, that's multiple voxel pattern analysis. It's cool, it's important, it's discovered many deep and fascinating things. With respect to the fusiform face area, it's a pain in the butt. We ran this experiment on a whole bunch of people, and we found exactly the same correlation between as within. I ran other experiments in my lab at the time. We found exactly the same correlation. I gave up on using MVPA in the fusiform face area, but we'll hear a paper shortly that got it to work in the fusiform face area. Other people have tried it. It's right near the edge of significance. You either just barely get it or you don't get it at all. In my hands, I didn't get it at all. In a few other hands, they just barely get it. But it does work better in other parts of the brain. Okay, so in the parahippocampal place area, you can really decode patterns quite more readily. You can tell if a scene is an outdoor scene or an indoor scene or other things like that using MVPA and the PPA. So I think the deal with MVPA is it works insofar as there are different patterns, right? But remember that each of these voxels shown abstractly here is maybe 500,000 neurons. Or in the case of this experiment, we scanned with the highest resolution we could. There were probably 100,000 neurons in there. Even so, it's 100,000 neurons, right? So if the neural code exists only at a fine grain of neuron to neuron and will totally wash out by the time you get 100,000 or a few 100,000 in a voxel, this just isn't going to work. So there's some parts of the brain where that information, where the neural code is actually clustered at a gross enough spatial scale that this method works and you can see information. But this is a classic case where when you don't see information with this method in a region, you have no idea. You certainly can't claim it's not there because it could easily be there at a fine grain. And there are places where we know from other research, well actually next time I'll talk about monkey neurophysiology, where we know that the identity codes are not spatially very organized in monkey face patches, making it unsurprising that this doesn't work in humans. Other places where we know this are in the hippocampus, where you have place cells that respond depending on where you are in space. And it's been shown over the last few years that place cells are each place cell is very, very specific to where an animal is in space, but there is no spatial regularity. So adjacent place cells are just as likely to respond when you're in totally different parts of space as when you're in similar parts of space. And so MVPA doesn't work in the hippocampus for space either. Okay? So when it works, it's lovely, and when it doesn't, it's just a bummer, and that's what it is. Okay. Okay, so just to wrap up, so all of that was just to say that we have 3 basic kinds of methods to use functional MRI to go beyond just saying, here's a region that's specialized for X, to try to say, what does it do about X? What does it care about X? One is just a simple one that's sometimes called univariate, where you just measure the response magnitude to a bunch of different stimuli that somehow manage to test your hypothesis, like moving versus snapshot of faces. That might give you a clue. Or the presence of external face features and face configurations, or the absence of it. The next one is functional MRI adaptation, where you exploit the higher neural response to 2 stimuli that are different compared to 2 stimuli that are the same, in order to ask each region of the brain whether it's sensitive to that particular difference. And the third one also asks whether a part of the brain is sensitive to a difference, but in this case you look at a whole pattern over a region of the brain, and you ask whether that pattern is stably different for 2 different kinds of stimuli. Okay, so now we're getting into we're getting closer to the cutting edge here, but also into the fiddly business where you have to mess around with scanning lots of subjects and being right near the edge of statistical significance with these methods. But on the other hand, they're worth it because these are really the questions you want to know, not just what is it specialized for, but what does it represent.